Greetings all, and welcome to the home of hard science and correct inferences. And today is St. Patrick's Day, so I'm wearing a kind of Zelensky green, if you will, because as you know, I'm into the latest thing, right? And Dr. John Campbell today in the UK, kudos to him because he's released key footage from the House Select Subcommittee on the viral origins. And this clip I'm going to show you now from his YouTube shows Dr. Redfield, who was former director of the CDC, no less. And Dr. Redfield was in all the conversations and emails with Fauci and Farrar and all the other Wellcome Trust people early on in coronavirus. But then he was excluded specifically by direction. So a subset of 11 people got together to create the Nature paper and all of the propaganda, which said it came from bats or raccoon dogs or whatever stuff they made up. So now we have proof of conspiracy as if we needed it, but it's delightful to watch it all come out in the House Select Subcommittee in the US. So have a look at this clip.
Dr. Redfield, you pointed to the lab leak theory even before we did. In mid-January of 2020, you expressed concerns to Dr. Fauci, to Jeremy Farrar of UK's Wellcome Trust, and to Dr. Tedros of World Health Organization that, quote, we had to take the lab leak hypothesis with extreme seriousness. And you urged Dr. Fauci to investigate both the lab and the natural hypotheses. Shortly thereafter, on February 1st, Farrar convened a meeting of a group of 11 top scientists across 5 time zones and asked Dr. Fauci to join, and he wrote, quote, my preference is to keep this group really tight. Obviously, ask everyone to treat in total confidence, unquote. Dr. Redfield, you were excluded from this call, but up until then, you had been on you were included in every other conversation. What changed? Why do you think that you were excluded from these conversations?
Thank you very much. I think just to emphasize, in early to mid-January, I did have multiple calls with Fauci, Farrar, and Tedros about how important I thought it was that science get engaged in aggressively pursuing both hypotheses. I also expressed, as a clinical virologist, that I felt it was not scientifically plausible that this virus went from a bat to humans and became one of the most infectious viruses that we have for humans.
Now, why do you think you were excluded from those calls?
Because it was told to me that they wanted a single narrative and that I obviously had a different point of view.
Okay. In emails following the conference call, 4 of the 11 scientists told Fauci that they all found the genetic sequence inconsistent with expectations from evolutionary theory, basically what you're saying. However, just 3 days later, these 4 scientists had drafted a paper arguing the exact opposite, and that's now the infamous proximal origin of SARS-CoV-2. Our investigations show that this paper was prompted by Dr. Fauci, among others, with a goal to disprove the lab leak theory. What is the likelihood that these scientists came across additional information just 3 days after making these statements to conclude with such certainty that COVID-19 came from nature instead of the lab leak that they thought it was 3 days earlier?
Yeah, I think it's unfortunate. Again, I've said this before, that this whole approach that was taken on February 1st and subsequently in the month of February, if you really want to be truthful, it's antithetical to science.
Thank you.
Science has debate, and they squashed any debate.
Thank you.
And there you have it. There is much more coming out in that committee, but that gives the gist of it. So bona fide, absolutely, conspiracy was underway, and it just matches all of the other instances of proven, documented, essentially conspiracy, not conspiracy theory, that occurred during the last 3 years. And I'm going to keep this short, so I won't show you all the other footage, but I'll put a link to Dr. Campbell's review below with more of the committee coverage. And now I'll just show you back in, I think it was May or June 2020, I had to take a video off this platform, even though it was entirely correct. And I think it's one of the best interviews. It's with Gabor Erdoshi, and it brings home the fact that overwhelming probability from 4 primary scientific vectors indicates that this was indeed gain and function, which is being explored in the committee, and that's just the way it is. And that actually would have driven this group of 11 who had to conspire together to squash that conceit or that idea. They had to, because essentially I would say they knew it was true and therefore they had to double down. So here's a short clip from myself and Gabor running through just the mouse development lines, gain of function evidence in the lab origins theory. And there are, as I say, 3 more primary vectors we go through. But just have a look at this clip, and then the full interview is down below on Odyssey. So here we go.
And then I was looking at a study, and there was a figure, columns showing the, I think 6-mer and 7-mer. It means how long peptides are identical, and the 6 or 7 peptides are identical between different animal proteins and the viral spike protein. And of course, there was a high similarity with humans because it spent a lot of time in humans. So you expect a certain level of mimicry developing, because then the virus can evade the immune system, avoid early recognition, and so on and so forth. Perhaps it was a bit too high already for humans, but what was really striking, right next to the human column, high column, the same height column was the mice. Musculus, the Latin name, put under the column mouse. And the mouse ACE2 receptor, which this virus uses for entry into the cells, is one of the worst receptors for the virus. So mice are absolutely not susceptible to this virus, unless there is a special type of mouse strain used in research, a kind of genetically modified mouse. It's called humanized because the human ACE2 receptor is expressed by the mice. So on the cell surfaces, they show the human receptor. And of course, the human receptor, the virus can bind very strongly and quickly. So this is a research tool in labs, and it was described, I don't know, 2013, 14, 15, so 6, 7, 8 years ago, how researchers used it in the US. And then this research was moved to China because there was a ban initiated in 2014, I think, in the US. This type of gain of function research in model animals, which try to mimic humans in some ways, was banned in the US. The financing funding was banned, was stopped, and this research was moved to China. But funnily enough, it was still financed by the US, mostly for some kind of circumventing this ban. And I think this one cannot be explained in any other way, how mice have the same level of peptide mimicry to do a viral spike as humans. It can only be explained by humanized, genetically modified lab mice. Otherwise, we are not aware of any. I haven't heard of any natural mouse strains having close similarity with human ACE2 receptor. So this is just striking. I mean, what was even more peculiar was how the authors went about it. But perhaps not the authors, but the reviewers and the editors. It always happens when, if you want to publish something, you have to accommodate to the expectations. So they point out that there is extreme level mimicry between humans, mouse, and viral spike, and then add no further comments whatsoever. The only comment is that mathematically this similarity is extremely improbable. I think that's the only comment what they did. I would love to see the original manuscript submitted and the exchange between the reviewers, editors, and the authors, because that would be really telling what happened. I can imagine that the original manuscript was a little bit different, pointing out that okay, this can have only one explanation because we cannot come up with anything else. Most likely they were silenced altogether.
Yeah, and again, not silenced in a conspiracy theory way, though. I guess, look, conspiracies are part of our modern world everywhere, and
whatever you call it. I mean, it's basic reasoning. You find some figures in a peer-reviewed journal article, and you try to make some sense of it. I mean, you theorize something or hypothesize something, because that's what you do when you see unexpected results. If you see unexpected results, you try to explain it. And if there is only one explanation that comes to mind one and after and again, then probably you are not wrong. Because, and I tried to ask several scientists openly on Twitter, for example, and they readily respond to other questions, but not to this one. So
Yeah,
if
and
I ask them, what do you think about this, they don't block me or anything like that. They just remain silent. They are not ready to take an open stance on this, because they have a career in academic and whatever, and probably they are worried that this could be something which puts them in the danger zone or whatever.
so there you are, folks. Hard science, the best kind. And do remember the links are down below to Dr. Campbell and, of course, to myself and Gabor Erdoshi's interview on Odyssey, which goes through all of the 4 primary vectors and constitutes essentially a form of proof, but I'll let you decide. Thanks so much, as always, to my Patreon supporters. I had another monthly Zoom last night with the business health patrons, and I also release vlogs and special insights as regularly as I can. So if you can support there, or indeed donation through PayPal, really appreciate it, and helps keep the investigative journalism, scientific, but also geopolitical and soon to come financial world investigation, which should help you and your family. So great stuff, folks, and till next time, happy St. Patrick's Day.